that, please take it away. Sadjad, thank you so much for agreeing.
Thank you very much for inviting me. So I'm going to talk about epithelial mechanics and how can we start understanding the mechanics of these complex systems in terms of learning and adaptation. I think everyone who works in biology or any biological systems, they have a favorite movie. This is my favorite movie in biology. This is basically the developmental aspects of an embryo, Drosophila embryo. And we can see every polygon is basically a cell. You can see the movements of this cell, the tissue flow, this kind of contraction, extension, all these different complex properties that is going on. So I'm really interested in understanding this basically problem of development in terms of adaptation and learning, and I'm going to talk about what I mean by that. So of course, we're living in the era of AI. Everyone is probably using ChatGPT or some version of these AI tools. Here is my summary of what I mean by learning in artificial systems. For example, if we want to do image recognition tasks, like image, basically a bunch of input, output, basically task here that we have a bunch of images, and we want to train these complex neural networks to get the right category. Usually we have this hidden layer that we can tune the properties of and the node weights of this hidden layer to basically train the system. In terms of biology and how I'm trying to connect this to tissues, is that this input-output relationship for me, for someone who's interested in tissues, that can be stimuli and response relationships. So in basically this artificial neural networks, usually you just write down a cost function. What is the learning cost function for this system, the error function? And that would be basically the actual output minus the predicted one squared, for example. And then you can go down in the gradient of this cost function by tuning some node weights. So we're basically using back propagation to go down in the cost function. And after some iterations, then the system learns, and basically you can differentiate between different images. But not only in artificial neural networks, in the past few years, people started to apply this type of tools and this type of algorithm in physical systems. So physical systems such as spring network. Here I'm showing an animation from actually Daniel Hexner lab that instead of neural networks, he's basically trying to tune the properties of disordered spring networks. And the task here, instead of image recognition here, the task is that you apply some input strain in some part of the network. And then the task is to get the same output strain, the same basically strain in different part of the network. How can you modify the learning degrees of freedom in this system to basically get to this task? Here I'm just citing some few recent papers on this topic, and I think it's an emerging kind of field that can be useful for soft matter as well as biophysics. So in this system, if we connect that to artificial neural networks, the learning degrees of freedom here, you can think of them spring stiffnesses, tuning the stiffnesses of these springs, as well as rest length. The cost function would be basically similar to what you can write down for ANNs. That would be just the desired strain minus the current strain in the output squared. And then, but this is a physical system. So you have physical degrees of freedom, which are these node positions, as well as a physical cost function, which that would be the energy. And so by just looking at this system, it's obvious we have these two landscapes. We have the physical landscape and the learning landscape. And the idea is that you can write down some algorithm to basically go down in the gradient of this learning cost function and get to desired task that you want. And that's what is plotted here, that the output strain is getting closer and closer to the input strain as the training goes on. But not only in spring networks, here I'm just showing some examples of this computation with physical systems that has been looked at from resistor networks here at Penn to computing with optical systems, as well as basically playing with the molecular concentration to do DNA self-assembly in a specific kind of setup. But these are all basically the computation with physical systems. And I'm interested in tissues, in real biological tissues. I think of tissues as these extraordinary soft materials. If I can compare these two, we start with non-living soft matter, like this gum wall in Seattle, actually, that just people put gums on the wall, and you can see the macroscopic behavior of this system, right? You can see this stretching behavior, that basically this is kind of the effect of gravity on these polymers. The microscopic structure of this system, if we look at it, is basically a bunch of long polymer chains. And you can tune the properties of the system by playing with the length of this polymer chain, as well as basically the mesh size of the system and the concentration as well. If we go then talk about tissues, the macroscopic properties that we see is basically this large deformation of these cells on the epithelium of this embryo, and this tissue flow. But the microscopic structure is very complex. The microscopic is not a simple polymer chain. You have basically active ingredients. Here, for example, showing myosin, actin, as well as catenanes that basically tune the properties of each cell. Catenanes helps to connect individual cells. Myosin and actin help to basically contract this cell edges. So by just comparing these two, we can see that these tissues are basically they have this microscopic structure that is complex, but also is tunable via different molecular concentration and proteins. So the idea is that since we have this setup already, these cells are actually tuning these properties already, and we know the macroscopic kind of effects as we just look at these tissues. Can we understand basically, and can we use these learning theories that people have used in AI, as well as these synthetic kind of materials, synthetic training of these materials? Can we use these learning theories for tissue mechanics? And if yes, then what are the adaptive degrees of freedom here? What are the spring stiffnesses or rest length, in a sense, in this setup? So to just start, here are some examples of tissues that I have in mind. These are examples of confluent biological tissues from MDCK monolayer. This is just a dog kidney sample, as well as this fruit fly embryo that I mentioned before, as well as this human bronchial tissue. All these tissues, they have some characteristics. They're confluent, so there is no gap between these cells. Every cell is connected to each other, as well as basically these are 2D layer of these tissues. And the reason for that is because these epithelial tissues are important for covering surfaces, for example, internal organs. And to start with focusing on the learning and adaptive degrees of freedom in the system, we need a model. So I'm just gonna talk about the first part of my talk, which is basically understanding rigidity transitions in tissues. So that would be the first story that I'm going to talk about. And then the second part of my talk, I'm going to talk about convergent extension and how can we understand convergent extension in Drosophila using this framework. Biological tissues, this epithelial tissues, known that they undergo rigidity transitions. Here I'm just citing some few papers out there that people have looked at this rigidity transition in tissues. Specifically, this paper from a few years ago showing this nice basically separation of solid-like state of these tissues and fluid-like state of these tissues by just looking at geometry. So looking at the shape of the tissue, the alignment, and looking at rearrangement rate of these cells as a proxy for rigidity, you can actually differentiate between solid and fluid-like states, and that can be explained using simple vertex type models. So this is the question that I'm interested. How can we understand rigidity of tissues in terms of this framework that I mentioned? To explain more this rigidity that has been observed in these tissues, here is basically a jamming-like phase diagram for biological tissues. We know, for example, fluctuations, the effect of fluctuations and density on the rigidity of these tissues. If we have a solid-like state, increasing fluctuations make it more fluid-like, or decreasing density as well. But for these specific tissues that I'm interested in, fluctuations can be negligible as well as basically density fixed, but still we can go through a rigidity transition. And that's just due to geometric incompatibility between basically parameter and target parameter that I'm going to talk about. And this geometric incompatibility is a fancy name for pre-stresses. So the system is basically pre-stressed in the solid state and stress-free in the fluid-like state. So if I plot the shear modulus of this system versus the target shape index, so this is a geometric factor of this tissue that I'm going to describe in a few slides. But then in this basically axis of this phase diagram, we go from a solid-like to a fluid-like state. Solid defined by finite shear modulus and fluid-like state defined by zero shear modulus. And the critical point for this transition happens to be around 3.9 in this space. Basically, the question that we want to ask is, how can we fluidize these tissues? If we start from a solid-like state, if we start from basically state of the tissue that's supposed to be solid-like, can I fluidize it to basically a floppy-like state? And that's important for many biological processes that I'm mentioning here, from crypt folding as well as these developmental processes in Drosophila. So which learning degrees of freedom are important to basically fluidize this system and get rid of these pre-stresses. To basically start with this answering this question, we need a model to explain basically the mechanics of the tissue. There are many great models that have been proposed in the literature from basically cellular POTS models for tissues as well as deformable particles. But I'm going to focus on these Vertex Voronoi type of models that has been successful to explain this rigidity transition and these properties of tissues in terms of this shape factor. In these Vertex type models, then we model every cell as a polygon with some vertices, with some random number of vertices. And the elastic energy for this system has two main contributions: area elasticity and perimeter elasticity. With A would be area and A0 would be target area, as well as area stiffness, perimeter and target perimeter. If we just expand this energy, then we find the first part is basically related to 3D incompressibility effects of these cells, as well as the p-squared term in the second term in the energy that's related to actomyosin contractility, the restoring force for that actomyosin ring, as well as the linear term that would be interfacial tension. So that's the model that I'm going to focus on. And to connect that to the original basically framework of learning, the vertex coordinates of each cell, these are the physical degrees of freedom, and we can think of this elastic energy as basically the physical cause function in that setup. As I mentioned before, we go through a rigidity transition versus a target shape factor. This target shape factor is nothing but a dimensionless parameter that comes out of that elastic energy and non-dimensionalizing that elastic energy. That basically is nothing but the ratio of target parameter over square root of target area. And we find that if we increase this target shape factor, we go from solid-like to fluid-like state, and the critical point is around 3.9. To basically explain the effect of this adaptive degrees of freedom on this rigidity transition, I would like to also quickly explain why this is happening, why we have this rigidity transition in these tissue models.
Sorry, can I ask real quick, this number 3.9, is that true across multiple systems or for a particular system?
This is basically true across multiple different systems. This is just a geometric shape factor. Like even if you model a tissue as deformable particles, you get to that state where everything becomes jammed, and that's close to 3.8 to 3.9. But that depends also on the amount of disorder you have in the system and the finite size effect. So it's not exactly 3.8 or 3.9, but it's in that basically region.
Yeah. Thank you.
Yeah. So to explain the nature of this rigidity transition, I'm going back to Maxwell's counting argument, which is really a simple, elegant counting argument that can explain rigidity of frames. For example, if you have 4 nodes and these 4 basically constraints, these 4 constraints on these nodes, then we can just count number of degrees of freedom in these 2D systems, number of constraints, remove the rigid body motions, and get one floppy mode. And that would be the floppy mode that I'm showing here. So the system is floppy or soft. Of course, we can get rid of this floppy mode if we just add another constraint in the system. So that's the simplest kind of way to check if a frame is rigid or floppy. This is not the whole story, of course. If we have these 2 snapshots of these 2 structures with the same number of nodes and the same number of constraints, the one on the left is actually rigid-like, and the one on the right is a floppy structure, and that's due to emergence of these states of self-stress, because we added this basically constraint on the same square here, so we introduced state of self-stress, and that's basically why we have this 0 mode in the system. So the basically the extension of this Maxwell's counting argument is that number of degrees of freedom minus number of constraints should give us number of 0 modes or floppy modes in the system minus number of states of self-stress. And that's really a simple way to check if the system is rigid or not, as in a first order rigidity basically problems. This simple counting argument can explain the jamming transition of particle packings, but actually vertex models are highly under constraint. So for example, if we just count number of degrees of freedom in these polygonal models, then we have 4 times number of cells as the degrees of freedom in 2D, a number of constraints, since we have 1 area constraint and 1 parameter constraint, and that's twice number of cells. But still we have this transition, right? If we have a tissue with P0 of 3.1 and compare that to a tissue of 3.96, definitely this is a rigid-like state. This is a floppy-like state if we just try to shear this system. And to really understand what's going on, we can look at edge tension. So between these cells connected to each other, we can compute what is the tension between those cells. And if I plot the edge tensions with the thickness proportional to the amount of that edge tension, and then I'm plotting basically shear modulus versus target shape factor, then we can see as we increase this shape factor, this large spanning cluster of edge tensions vanishes at that critical point. Basically, we get a more floppy-like state without any spanning cluster of edge tension. So that's a tension-driven kind of rigidity related to the pre-stress in the system.
Sorry, can I interject here for a second? So that's something I always wonder about in like this particular area perimeter energy with the tensions vanishing, right? So this state of self-stress disappearing. If I look at the tissue and I do a laser ablation experiment, even in the regime where the cell shapes are this very disordered, floppy cell shapes, the individual junctions still spring back. They're still, so I would say, like experimentally, there's evidence they're still under tension. So isn't that somehow in contrast to what this model says?
That's a great point. So one thing that I would say is this is basically, as I mentioned, where the rigidity transition is definitely not only depends on the amount of disorder in the system, but also depends on the anisotropy of your tissue also. So if I change my tissue to be more kind of rectangle, if I just stretch it, then of course I'm going to change where the rigidity transition point is, although the P0 can be 4.1. So the P0 definitely is one indication of where the rigidity transition is, but it's not the whole story, because anisotropy of the tissue also changes where these edge tensions are and where they're going to vanish. And that's actually related to that 2020 PNAS paper by Karen Keiza and Lisa Manning, showing that you need 2 axes to explain the data. You need the anisotropy and the alignment of these cells as well as the shape. So I would say that's a great point, but I don't see a real contradiction here because that depends on the other factor as well.
Sorry, I mean, I know this is not like the main discussion point or the main point you want to make in your talk, but just to reiterate, right? In Drosophila, whenever you do a laser ablation, even later on when you are in that picture in Karen's paper above the fluidity transition, there's still, all the junctions are under tension. They all recoil.
That's a good point. Maybe we can talk.
It's just something I always wonder about when the Vertex model is discussed. So, but sorry, I mean,
no, that's a great point. I would like to talk about this later if that's okay, since I'm gonna go through the slides, but that's really a great point. And so basically, I talked about this rigidity transition versus this shape factor as one parameter, but there are some basically nice work out there that shows not only the average shape factor is important, but also the anisotropy of these shape factors as well is important. So related maybe to the question as well, the heterogeneity of these P0 values also can change where this rigidity transition is. And that's from a nice paper by Max B and others from a few years ago. In this plot, I'm showing the shear modulus versus the average target shape factor by increasing basically the standard deviation of this shape factor. So if we add a heterogeneous kind of shape factor for this tissue, and then just increase the standard deviation of this normal distribution, we can see that we can tune where the rigidity transition is, basically by increasing this standard deviation. So since we know not only the average, but also the fluctuations of these shape factors are important, the question is, if I just add these shape factors or stiffnesses as new degrees of freedom, and I let cells to tune these variables, then the question is, can I use these new variables to actually fluidize the system? If I start from a solid-like state at the same basically distribution of shape factors, and then I just basically use these new degrees of freedom to let them tune and change spatially, can I actually fluidize the system and go to a zero shear modulus state? So in these vertex models, the learning degrees of freedom, we can think about them as area stiffness, perimeter stiffness, target areas, and target perimeter as variables that we can think of as adaptive degrees of freedom or learning degrees of freedom. And this is actually related to a really nice work by Vardahar a few years ago that they looked at the effect of new degrees of freedom in their case for packings of spheres, and looking at the jamming of that system. They found that stiffnesses and radius, they have different effect on the jamming of that system. So the question is, what is the effect of this new added learning degrees of freedom on the rigidity of these biological tissues? This is my one slide on the method, so please ask questions if something is unclear how we're doing this. But we just start with a random tissue, and I would like basically to start focusing on one of these parameters, for example, area stiffness or in target areas or KP. I want to add one of these new variables for these cells as adaptive degrees of freedom, let that to be tunable within a distribution. So I would like to fix the distribution with basically constraining some moments of that distribution. So if I add KP as new degree of freedom with a normal distribution, I also do constraint minimization to make sure that actually the distribution of those KP values stays fixed during all these optimization problems. The reason for fixing the distribution is because as I talked about, changing the rigidity by changing the distribution is an easy task. That's already known. But the question is, can this tissue actually change the rigidity by just spatial correlations that appear because of this optimization problem? So here I'm just showing basically the results of the shear modulus versus target shape index by adding stiffnesses, by adding either area or perimeter stiffnesses as new degrees of freedom. The black one is the usual scenario, the only physical degrees of freedom, and the colored one are either for adding area stiffnesses or perimeter stiffnesses. In this case, since I focus on stiffnesses, this target shape index is uniform. So this is the same for every cell. We can see that although you can get slightly softer tissue, actually you cannot tune where the rigidity transition is. So it's actually not, these stiffnesses aren't important for tuning where this rigidity transition is in this set.
Sadjad, can I ask a quick clarifying question just to make sure I'm following? The minimization you're doing is of the same, with just the energy functional, right? You're just now minimizing it over more degrees of freedom.
Exactly. So here, basically my physical cost function, learning cost function, they're both the same because I'm interested in rigidity. So I want to fluidize the system.
Yes. But are there situations or have you thought of cases where one may want to separate the two, because it's potentially by minimizing the 2 degrees of freedom simultaneously, then it's a statement of time scales, right? So you're saying that both of them equilibrate simultaneously. But are there cases of interest, either you'll have looked at or of experimental interest, where the physical degrees of freedom may equilibrate faster and the learning degrees of freedom may be much slower, in which case you would have to decouple the two?
Exactly, that's a great question. So in the second part of my talk, I come to a second basically process where these two actually cost function are different.
Okay.
And then I talk about how I'm tuning these variables, these new variables, to go down in a different cost function than the energy. It is true, even I would say, even for minimizing this elastic energy by adding everything, still more stable to actually minimize the physical first and then add the new degrees of freedom. So have a separate kind of time scale as well in that case to have a stable solution. So your basically insight is really correct, that these two kind of sets of degrees of freedom, that they have different time scales. But in the second part of my talk, I'll talk about a different cost function as well.
Okay, thank you.
So stiffnesses actually don't matter. Basically, they can't tune the rigidity transition point. So then we focused on these target shapes, these P0 values. And we can look at the shear modulus versus here. Now it's going to be the average target shape, because these target shapes are distribution now. It's a normal distribution that stays fixed in this optimization problem. And then basically the black one is the usual scenario. The red one is after adding these shape factors. And we can see that by just letting cells to tune these shape factors, we can tune the rigidity transition point. We can go basically lower in this space and fluidize the system at a lower shape factor, basically. I.e., if I start with a solid-like state and let cells to tune their P0 values, I can actually fluidize the system with the same distribution of shape factors. So the distribution didn't change. I just changed the spatial correlation in the system. This same kind of effect can be observed if I add these target areas. So that's also can tune the rigidity transition point in the same way. So I want to also explain briefly why this is happening. For that, I'm just gonna show 2 snapshots of these tissues. So this is shear modulus versus average target shapes. The black one is the usual scenario. Nothing is added. It's just the vertices. We're just minimizing the vertices, the elastic energy with respect to vertices. And the green one is after adding these shape factors. These 2 snapshots of the tissues, the one on the left is the usual scenario. The one on the right is after adding these shape factors as new degrees of freedom. And they're colored with their shape factors. So although the distribution is spatially different, but they're basically the same exact Gaussian distribution here. By just looking at these 2 snapshots, it's hard to see why we have a floppy state. But since this is related to edge tensions, I'm just gonna plot the edge tensions on top of these cells. And you can see that on the left, we have this cluster of large edge tensions that is responsible for rigidity of this tissue. And on the right, basically the system smartly got rid of that cluster by just tuning where these P0 values are. They basically got rid of this large spanning cluster. And that's why the system is floppy, although we have these local edge tensions as well.
Also, Sadjad, another question. Is Is this a common feature that when you have these learning degrees of freedoms that the system sort of organizes itself or sort of minimizes until you hit the transition point? Is there a version of sort of self-organized criticality that one should think of over here?
So I can't really say if this is a common feature. What I can say is, well, for example, if we start with a distribution that is right here, of course, we can basically get rid of some tensions on these edges, but they can't really get rid of the rigidity. So although they have a different spatial distribution of these P0 values, but still the system is rigid with a lower shear modulus. So it's not always going to basically fluidize. So there is a region, right? There is a region where I can actually fluidize the system if I start with a distribution close to that.
I see. Okay, so it isn't as if that if you started with a different distribution and you reran and reinitialize the whole minimization there are distributions of edge stiffnesses and all of these parameter the perimeter area ratios and whatnot, the preferred perimeter area ratios. There are distributions of these parameters for which you cannot fluidize the tissue, even with the learning degrees of freedom being minimized upon.
Exactly. Yes, that's definitely correct. And the reason for that is because I'm constraining the distribution, right? I don't let the distribution to change. But also it depends on how much fluctuations I allow, right? So
that's the width of the distribution you're talking about, right?
Exactly. So in the next slide, I'm just going to talk about the effect of the standard deviation of this distribution on this basically shift in the rigidity point.
And you're only looking at ground states. You're not looking at dynamics. Is that correct?
That is correct. So I'm only looking at the ground state by just the minimum energy state.
Okay, thank you.
Yeah. So exactly, this was a great question, because in this slide, I'm just going to talk about basically the effect of the standard deviation of the distribution, this P0 distribution, on how much I can tune this rigidity transition point. So the purple line is basically the amount of the shift in the transition point. And we can see there is basically this non-monotonic dependence on how much we let these P0 values to fluctuate in the system, and how much we can shift the rigidity transition point. And we can see that there is basically an optimum value of this standard deviation of these distributions around 0.15 that gives us the highest basically change in the rigidity transition point, and shifting the rigidity transition point, but it's actually going down if we just let everything to be basically if we just increase the width to higher values. So there are, I didn't talk about the structural features that emerges and how this basically adding these adaptive degrees of freedom is going to change the structure of these tissues. For example, the number of hexagonal cells increases when you let cells to tune their adaptive degrees of freedom. But for the sake of time, all those details are in our preprint, and basically talking about the structure and the vibrational density of these systems with and without degrees of freedom. But if there is no more questions, I'm actually going to change gears slightly and talk about the second part of my talk. So in the second part, I'm going to talk about how can we study convergent extension in this system, convergent extension in Drosophila in terms of a learning problem. All I talked about in the previous part was the rigidity transition. How can I shift the rigidity transition by adding these new variables as degrees of freedom? But here I would like to actually get this kind of flow. So convergent extension here I'm showing an embryo of Drosophila, head and tail, and you can see that after some time, this yellow line is basically a visual aid showing that this tissue becomes elongated, almost twice its size, in basically this axis, and contracts in the basically the y-axis on this page here, and then basically folds over itself. So this process takes about 2 hours, and that's an important part of this development of this Drosophila. Can we understand this, and can we get this tissue flow using this framework? To just mention where that process is in the developmental cycle of fruit fly. Here I'm just showing the developmental clock of this insect. That's the elongation of the germ band starts at the end of stage 7, and that's gonna start basically just elongating and folding. That's gonna take about 2 hours to finish. So just thinking about the macroscopic deformation that happens in this system, if I just focus on a square of that tissue, is actually after some time, it's gonna double its length and contracts in the y direction. That's basically the macroscopic deformation that we see. If you think about mechanisms that could be responsible for these, schematically, we can think about cell rearrangements, right? How cells can rearrange themselves to go from a structure like this to a more elongated structure like that. That's going to increase the length of the tissue and contracts in the y direction here. As well as stretching, right? If you just stretch this tissue, you can also increase this aspect ratio. So how can we understand this process? I start with the same basically vertex models, with the usual basically vertex model that I talked about. But to be able to actually add tensions, edge tensions as new degrees of freedom. So I would like to be able to play with edge tensions as degrees of freedom, to be able to tune those variables instead of just passive edge tensions that comes out of the usual vertex model. I'm gonna add a new term. That's basically the tension energy. This is a tension on each edge, and this is the length of each edge. And that's the additional tensional energy that I add in the system. And I would like to make this basically this process of shear deformation, this pure shear deformation, I would like to make it soft. How can I make this process soft in my tissue? How can I tune these edge tensions, these additional degrees of freedom, to make this kind of process to be the soft kind of direction in the energy landscape? To do that, what I did is basically just taking the gradients of this tensional energy on each edge with respect to the deformation. I would like to make this a soft direction. So I would take the gradient with respect to that deformation and go down in that gradient locally for each edge. What comes out of that is basically depends on the orientation of each edge. So what we need to do is basically saying that I'm going to increase the tension that are oriented vertically and decrease the tension that are oriented horizontally. That's going to help to basically get us to this aspect ratio that we want. So that would be the rule that I'm going to follow in this system. That's local rule for each edge. I'm going to change this tension locally based on the orientation. And I start with just a random energy minimized tissue, the usual scenario here. I initialize the edge tensions with the passive edge tensions and that I get from the usual vertex model, and then just start this tuning of these edge tensions based on this rule. So I'm just basically changing edge tensions based on their orientation. So here is what we find. Here I'm plotting edge tensions. Brighter color means larger edge tension. And here I'm plotting the aspect ratio of the simulation box, LX or LY, as I'm tuning those basically locally tuning those edge tensions. That by just this locally tuning those edge tensions, actually you get this sort of flow, this convergent extension flow, and the system is basically elongating. But also you can see these stripes of edge tensions that are oriented vertically. That comes out of just doing those local tuning of edge tensions.
Sajjad, a quick question. Are T1 transitions permitted or are they not allowed in these minimizations?
These are actually permitted, and you can see some of them, although it's fast to see which cells. I'm going to show some statistics of them and how that's related to this elongation.
And the convergent extension is, maybe you'll say a little bit about how much of the convergent extension is because of an isotropic shape change of individual cells. So cell-based shear versus T1 induced shear.
Yeah, I don't, I'm not gonna exactly talk about that, but I'm gonna compare this process with the external pure shear. So in the end, I'm gonna compare these two mechanisms, internally driven pure shear and just externally pulling and pushing the tissue, and how can we differentiate between these two.
Okay, thank you.
Yeah.
Sorry, I have a quick question here about force balance. So in the final state, we see a lot of these vertically oriented junctions that are under high tension, but then the angles of the two neighboring junctions are still quite obtuse. So if I think about like a rope that is under high tension or rubber band, and then I have two others at the sides, I would expect that the angle becomes more acute because of that differential in tensions, right? So right in the middle here, for instance, there's like these very short high tension edges, but then the neighbors form an obtuse angle. So I'm a little confused how that works with force balance. So what is the exact minimization scheme that you run here? It's still minimizing energy, so it should be in force balance, right?
Everything is at force balance right now. Although I understand your question, I think basically, I don't know exactly which edge and which setup you're talking about, but everything is basically at mechanical balance. The only difference here is that we're allowing this box, this shear box of degrees of freedom, to also be part of the energy minimization. So that's why we get this elongation, basically. But everything is basically at mechanical equilibrium. The only difference is that we have these edge tensions that are tuned internally.
Yeah.
Maybe a quick clarification there on the same context. So the colors you're plotting are which edge? Are they the edge tensions
good. That's a good
or
point.
are they the full tension that includes the passive one as well?
That was a good point. Yes, so these are actually just the active edge tensions that I'm.
So then it should answer.
Thank you. That I think that solves the problem. Yeah.
Yeah. So that's a good point. These are the active edge tensions. I'm not adding the passive edge tensions on top of this. And this kind of stripes of these edge tensions, that's also consistent with the classic understanding of why basically we get this tissue flow in Drosophila, going back to this nice paper from 2004, showing the distribution of myosin. So this is the myosin distribution versus the orientation, and the red one is higher myosin concentration. You can see it's basically on the edges that are oriented vertically, you have larger basically myosin concentration. That's consistent with this stripe-like patterns that we get by adding this local rule. But related to also the Suraj's question, if we just look at some samples of the system, we get those snapping points. So it's elastically increasing this aspect ratio, but also sometimes we get these snapping points. Those are actually the T1 events. So those are the T1 events that helps to increase this aspect ratio. And the T1 is nothing but basically you start with a 4-cell configuration like this, and then you change the neighbors in this confluent tissue to basically, and that's why it's gonna help this aspect ratio, because we get a larger edge length in the horizontal axis. And those are the snapping points that we see in this type of models. I don't think I have a lot of time, so I'm gonna go through this faster. If we also look at what kind of T1 we get, all these T1 events that we get are the one that are oriented vertically. So the edge, this is a summary of what exactly is happening. We start with really high tension on these short edges that are oriented in a vertical axis. And then this is basically, after a T1, is going to help the elongation of this tissue. And just related to other question, how can we compare this to an externally driven kind of deformation? If instead of this internally tuning these edge tensions, if I just stretch this tissue, what kind of edge tension patterns I see. So these are, this is basically a simulation of pure shear external deformation on the same type of tissue here versus strain, since we're just applying this pure shear deformation. To compare these two mechanisms, one way to compare them is just look at the average tissue shape, right? If I just look at the average tissue shape versus the strain in the tissue, the green one is the externally driven kind of deformation that we're just basically apply this pure shear. The orange one is this local kind of rule by just tuning these active edge tensions, that we can see that's the shape of the tissue is actually increasing in a higher rate compared to the passive or external case. Here on the right, I'm showing some experimental results of tissue shape versus here time, not a strain. That's during this convergent extension is increasing as the tissue is basically getting elongated. So this is an ongoing work to compare these two different scenarios, basically to real experiments. And I think this type of models of local rule, external deformations, as well as gradient descent. So the other part of this problem is that I can't just do everything as a gradient descent and tune those tensions to go down in the gradient descent of a learning cost function that would be this specific kind of aspect ratio that I want. So comparing these three different methods with experimental data, we hope to find or differentiate between mechanisms in this process. So with that, I'm just going to summarize basically what I talked about. I basically talked about that these cells have ability to tune their properties, shape factors, tensions, all these properties, and microscopically. This huge space of adaptive degrees of freedom is really a rich space to study. We can tune these properties, we can define the cost function, and look for local rules. Many of these processes involves this rigidity fluidity transition. We show that can be tuned based on what degrees of freedom we can allow to change. And in general, I think this framework, this idea of physical learning and looking at the system in terms of learning, writing down a cost function and looking for local rules can be useful to explain or maybe even discover new local rules in these systems. With that, I'm just going to thank everyone from Lisa's group, Andrea's group, as well as Indrajit, who's been influential for the first part of my talk, talking about the rigidity and fluidity in tissues. And thank you for your attention. I'd be happy to take any more questions.
Okay, please join me in thanking Sadjad for a wonderful talk. Suraj had to leave, so I'll be emceeing our question session, and the floor is open if you'd like to unmute and ask a question, or you're welcome to type it in the chat and I'll try and read it out. And I'll start us off with maybe a high-level question of what is learning and why is this learning? I understand that it is, you have minimization on a fast time scale that's just a force balance, and then you have a slower time scale minimization of some other cost function over the degrees of freedom, over the sort of parameters of the model. But when I think of the word learning, I think of a way of associating inputs with outputs. And so is this system doing that?
That's really a great point. And I think, so the way that I think about that learning, of course, there is no generalization in this system, right? So that's usually when you think about learning also in the context of AI, you need a generalization. But in this context, I think about learning and adaptation. I use these words interchangeably. That for me, it just means that I have this stimuli, this microscopic effects on each cell that are doing something, and the macroscopic or the output that I get. And for example, for this tuning of its edge tensions, I'm just doing that locally, right? For each edge tensions, I'm just tuning based on where that orientation is. And then you get this microscopic kind of output. So this is how I'm trying to understand tissues in terms of this locally microscopic stuff that are happening and this macroscopic emergent phenomena. So maybe it's a more kind of a general learning that I have in mind in terms of just stimuli and response. But I think a useful kind of lens, at least for me, that when I think about different processes, I can differentiate. These are physical degrees of freedom. This is the cost function that I want to have, what kind of rule that I need to find or comes out of this process. So that's the part that I think is useful.
Great, okay. Other questions?
So I was wondering what the rigidity transition, what role it plays in the second half of your talk.
Yeah, that's a great point. So here, basically, actually, we start in the solid-like state of the system, and we're tuning these edge tensions totally in the solid-like state, but we're actively inducing these T-bonds as well as this deformation by where those active edge tensions are. In this specific process, you can't actually tune it in the fluid side of the transition, because then you don't have those edge tensions to play with. So you need to tune these edge tensions where the system actually has this freedom to go between these different type of edge tensions. So it's right now it's an ongoing project, so I didn't look through all the different P0 effects. If I get closer to transition, if I get away, so I don't really exactly know what is the effect on that, on, for example, the shape, the rate of the shape, and things like that. But that's an ongoing effort.
Hey, can I ask a question?
Yes.
Great talk, Sadjad.
Oh,
Hello?
sorry, yeah, I can go later.
Oh,
All right, let's, Naki and then Susan.
Okay.
yeah. So I'm wondering, you talked about the learning rule that helps you get anisotropy in these tissues, and it seems like your learning rule contains, it is comparing to some angle, right? You're looking for some angle which dictates how each particular cell should behave. Now, what I thought is that this angle is some extra information that needs to be transported to every cell, which I can imagine to be sort of a global, like a global signal. And I'm wondering whether there are known ways in which tissues like that can submit global cells to everywhere in the tissue.
Yeah, that's really a great question, Aki. So that's true that the angle information is a global information. That's why we're also doing gradient descent instead of just this local rule based on angle to compare with this local rule, right? But for in the biology context, as I showed, it is, I think at least it's everyone agrees that the myosin is actually concentrated on these specific oriented edges, myosin and actin, and those are responsible for these edge tensions, right? So there is this, although you can also ask why, right? So you can go down in understanding the signaling pathways that are responsible why myosin is there and why actin is there. But for us, this is consistent with what tissue is doing in this stage, that myosin is actually getting concentrated on this specific type of oriented edges. And that's consistent with this rule that the tension is increasing on those specific type of edges. But on the modeling part, we're actually also comparing that to just gradient descent. No local rule, no edge orientation information, just doing low gradient descent on this cost function, the layering cost function, and then compare these three different methods, which are totally different way of getting this basically output, and which one is it, right? Which one is the way that the tissue is behaving?
Okay, but before we get to Susan's question, I had forgotten that it is our tradition to turn off the recording at a certain time, not to encourage the end of questions, but rather to encourage more questions, people who might not want to ask on the recording. So we will end the recording now. To people watching this in the future, thanks for joining, and stop recording.